Okay, thank you everyone for attending the Spatial Omics Seminar Series 6. And today we are so delighted to have Dr. Brian Fritz from Spatial Genomics to give a seminar entitled Advancing Biology with Spatial Genomics. So very brief bio about Brian. And so Brian got his PhD from Wisconsin Madison in molecular biochemistry or biomolecular chemistry. And afterwards, he moved to Seattle and started as a postdoc at the Fritz Cancer Center. And since then, I think Brian just told me he just loved Seattle so much and stayed there until now. And
most of the time.
Yeah, most of the time. And after Fritz, he joined the biotech industry and started his industry career at Illumina, I believe, and then Adaptive, and then moved to 10x Genomics. And what is terrific today is he can see lots of 10x colleagues here in the audience. And earlier this year, and he moved out of sort of the big biotech company space and joined Spatial Genomics and to help advance the technology based on the sequential phase to look at the genome-wide, transcriptome-wide gene expression directly in the tissue. So without further ado, and Brian, the floor is yours.
Okay, thanks, Rong. Again, thanks to you and Ahmet for the introduction to speak in this session. It's a privilege to join the ranks of the party assembled. So let's get into it. Let's have to get my first slide to go. Here we go. So, I mean, we're all here. By the way, I'm not going to show the nature method of the year slide this time. I figure we've all seen it enough. That's my one joke for the presentation, by the way. So we'll skip ahead to why we're all here in a different way, which is that biology is inherently spatial, meaning most of the biology that we're studying is able to be observed by microscopy or other means in 2 or 3 dimensions. And it's really at that level of the analysis that we started as biologists many generations ago. It's sort of always been with us, but of course now that those of us who've, like myself, wandered through genomics now for many years, we're sort of bringing our genomics methodologies into imaging. We'll touch on that a bit more throughout the presentation. And I think it's this really fascinating place in biological study that we're ambitious in this room, all of us here, to try and assay. We're interested in understanding not just the organization of bodies and organ systems. We've been using single cell analysis now for many years to do a much more effective and thorough job of cataloging cellular states and characterizing the distributions of those states within different types of tissue. But I think what those of us in this room are most interested in is understanding what that really looks like inside an intact tissue, because it's that space between cells and organs where we probably know the least in this entire spectrum, from molecules up to whole organisms. It's that piece of tissue that's the least well understood. And it's really the whole ambition of things like the Human Cell Atlas. It isn't just simply to count cells, but to know where they are and how the different tissues that they assemble into are constructed at a cellular, and I will propose today even a little bit at the subcellular level. So within our system of study then, the technology that I'll talk to you about, it's really an ambition to take sort of molecular genomics and imaging techniques and enable those to ask and answer some of these questions about the ways in which single cells are organized within intact tissue as a way of better understanding whole organisms and organ systems. And along the way, and again, I don't think these opening slides are dramatically surprising to anyone in this audience, but I'm going to say them anyway. I think some of the things that we can start to capture with these types of methodologies are, again, that catalog of cell states, but now with an added spatial dimension to them, and the ability to understand within those tissues and organs how cells are assembled and how they're organized and how they signal between each other. For example, we'll have one brief slide on this later in the presentation. And also towards the very end, I think we'll have time to talk about some of the developmental ways in which this can be applied in model organisms and potentially also in humans, where we can ask and answer questions about cell fate decisions, not in a dish necessarily, but inside tissue sections themselves. And again, as I've alluded to, we owe a lot to the technologies that come before us. So obviously spatial genomics, as we know of it, both as a company and as an industry, are really dependent upon imaging techniques. We're asking and answering questions about biology that we can see with a microscope. But in order to do that, the methodology that we employ here, and I'm sure others in this room do as well, requires a lot of prior knowledge from genomics generally. So the annotation of reference transcriptomic data sets are crucially important for us to do our work. And a lot of the methodologies that have been developed for a single cell analysis, particularly the computational ones in many cases, become really important for us to be able to make sense of the data that we're generating, because it is truly single cell data. Despite it having a spatial component to it, we can still think of it in that sort of counting of cells kinds of ways before we also then apply that kind of methodology to assaying the intact tissue and the locations of cells themselves. So really what we're trying to do, and we'll come through the presentation to at least the way that this technology at Spatial Genomics, which we call SeqFish, has been developed, is to be able to take intact tissue from a source. In this case, we're using model organism and a brain from mouse as an example, to take a slice of that tissue to resolve individual cells, but then to even gain a super resolve view of subcellular locations of gene expression in this case. We'll also look at DNA and protein throughout the presentation as well. And that collection of information is really what's informing our map of the tissue. It's not just a macroscopic view of tissue, it's a microscopic and subcellular view of tissue in a way that I think is really beautiful and fascinating about this data type. So who are we? Very, very briefly, just to say we're a young company, still very small, just a few dozens of us right now, founded almost 5 years ago. And again, with this interest of commercializing a spatial analysis technology suite that's called SeqFish. And we'll get right into it. Where does SeqFish derive from? Well, if we go back in time before SeqFish, this was published work by Andrew Femino and colleagues in Robert Singer's laboratory and Arjun Raj, then still a postdoc. And this is a little over 10 years ago already. Arjun's paper is sort of exemplified here as one way of thinking about single molecule fluorescence in situ hybridization. So the capability to resolve individual molecules of genes within an intact fixed tissue. The advance that Arjun and others made was to use multiple different oligonucleotide probes, schematically shown in the middle here. And each of these probes is conjugated with a fluorescent label. In the accumulation of that different number of signals all in the same color channel allows us to actually resolve the single molecule of the gene that's expressed in a particular cell. On the right-hand side, we have 3 images from Arjun's paper, actually, with 3 different genes represented. And we already here start to get a sense for what the data type looks like, as well as some of the challenges and opportunities of single molecule fish and detection of genes. And it's partially based on gene expression. So the lowest panel there, the KBP5 as a gene example, there's already a lot of gene expression in this particular cell state. And we can imagine now, if we wanted to extend this type of an assay to many hundreds or thousands of genes, if we simply use this type of approach, A, we'd run out of fluorophores really quickly, and B, we'd start to run out of space, because cells are very optically crowded places. They're full of genes and other things that are going to be fluorescing if we choose to detect them in this fashion. And so we have to separate out the signals from one another. And there's a variety of different methods that have been proposed to do this. I'll show you ours in just a moment. But really, it's a method that's going to enable us to take each individual gene, if we want, and image it by itself in the absence of any other genetic information being interrogated in the same image, and then to combine all those images together into a single super resolved version of that entire data set in an intact cell or tissue section. So here's just a very small number of the publication record of SeqFish and its technological development, its application to different biological questions of interest. We won't have time to touch on all these today. We'll go through as many as we can, starting with one of the earlier ones. This is from Long Kai's laboratory with the first version of this type of technology, again, published. And that was, again, as a reminder to solve a couple different questions. One of the optical crowding of different genes inside an intact cellular space, and to advance multiplexing capabilities. So we can actually look at more than 3 or 5 things, depending on the limitations of fluorophores that are available to us. So we'll talk about a barcoding scheme here. It's not the final one that we're using today, but it was just an idea at the time that gives us some sense for what's possible by using the standard set of fluorophores to which we're accustomed to using for these types of single molecule FISH assays, but doing so over a series of different rounds of imaging. And if we have, for example, a 4-fluor system and many different rounds shown here on the lower middle section, you get a sense for the scale of the barcoding and the multiplexity of gene detection that's possible with this type of a system. And here, if we're taking sort of the middle example, 4 fluors and 6 rounds, we can potentially resolve a little bit more than 4,000 different genes. But we start to see already in this schematic example, the coincidence of different genes with the same fluor in the same round of barcoding. So there is probably still an inherent gene expression problem that we're going to have to solve here in terms of optical crowding, but the multiplexing capacity is already dramatically improved relative to the original single molecule FISH publications. So if we step back and do a very simple version of this, 4 colors and 3 rounds, and schematically think about what that looks like in single cells, of course, we have genes expressed in a single cell and shown schematically in color here. We can change the fluorophore from cycle to cycle of imaging and collect a series of barcoded spots that change in color, but are in the same XY location. And when we see these different colors that we've already pre-assigned to genes light up in a certain pattern, we know that we can assign that barcoded signal back to a gene of interest in the data set. And that's in what this data type looks like in effect in microscopy image shown here in cultured cells as an example. We get a sense for what the data type looks like. And we get a sense for the ability to resolve genes one from the other in an XY coordinate system. We can apply this in a slightly more interesting way, not just to cultured cells, but to slices of mouse hippocampus. In this particular example, again, it's a relatively modest numbers of genes at this point a couple of years ago relative to what we can do today, but it was a really beautiful demonstration of the type of technology with this particular type of barcoding scheme at the time, 125 genes and about 15,000 cells that they counted in 2 coronal sections from mice. This was a 5-fluor, 4 barcode round imaging set. So it actually gives us 625 total barcodes. We're only occupying a fifth of that. So it gives us a lot of space in which we could, or they could, I wasn't involved at the time, obviously, in which they could understand off-target effects, for example. And even then it was obvious based on, again, gene expression differences, the amount of transcripts could make some of these difficult to barcode together. So they were able to barcode 100 of these genes together into various different image cycles of hybridization and imaging, but then 25 of those genes were high enough abundance that they had to be measured individually. And so you can still stack those together, 5 different colors, just gives you 5 different genes at a time over 5 serial hybridizations to achieve those 25 serial gene expression measurements. And when you apply that to a slice of the hippocampus, looking at different regions of that here, and you get a sense for the complexity of the data type. Even with 125 genes, we can do a very crude clustering analysis and define a number of different cellular subsets that you can then map back onto the image of the tissue itself. And you get a sense for the diversity of cells within those tissue spaces, as well as the distribution of those cells along, in this case, the dorsal ventral axis of the hippocampus, where there's relatively homogeneous gene expression and cell presence in the dorsal portion of the hippocampus, and it gets more heterogeneous as you move towards the ventral side. We're not really dwelling on the biology here too deeply. In case you hadn't noticed, we have a lot of ground to cover and not a lot of time. So technically, this is going to be more of the presentation is just focused on these kinds of technical improvements and details. In a subsequent work, and this was published a few years ago in collaboration with Guocheng Yuan, who I did notice is on the call today, so he might jump in and correct me if I say something particularly egregious. But I think the really beautiful part of this work is shown here, although there's plenty of good data in this paper. With even a cursory clustering of single cell RNA-seq data, and that's shown on the left-hand column under cell type in the middle of the panel. We know that there are certain types of neurons present in this particular section of tissue, and they're organized into various layers as the brain is organized, of course. And that's fine up to a point, but when you look at the actual spatial domains of gene expression, meaning that you look at the types of gene expression profiles that actually map to different domains, not just different cells, you can actually see there's a subtle difference in gene expression, even within the glutaminergic neurons, for example, in the top panel there. So even though they're the same cell type in a canonical sense, there's actually a little bit more detail that can be resolved by understanding their physical locations within an intact tissue section. And of course, we would probably see this in the data if we looked at a UMAP or a t-SNE plot, if we clustered it well enough. But we still miss that crucial piece of information about where those particular cells are in intact tissue relative to one another. And I think that's inherently the beauty of spatial analysis of this type. You get a sense for where these cells actually are. So it gives us the where as well as the what, so to speak. And so stepping forward in time, one more publication and thinking about how to solve some of the remaining questions of optical density. If we really want to get to a full transcriptome or even something approximating that, how do we do that? Because we start to run out of optical space with just barcoded probes that we change color in different sequence throughout the course of an experiment. Here in this more advanced version of the barcoding technique, and this is just one among many, obviously, we change up the scheme a little bit. So instead of having a primary oligonucleotide probe set, which binds to a gene of interest that's itself conjugated with fluorophores, we have a sort of a bipartite analysis instead. There's still a primary probe, it still binds to the targets of interest. There's still a number of them, as Arjun published many years ago. But now we have a series of what we call decode positions along the primary probe sequence itself. And you can occupy any number of these different sequences with a secondary probe that now bears the fluorophore directly. And you can flow these secondary probes into the image in sequence, hybridize them, image them, and then remove them, and repeat that as many times as you like to have as large a barcoding set as you need to resolve the number of genes that you're interested in detecting. In this case, the probes targeted over 10,000 different genes of interest, and the data is shown in the upper middle panel there. Some of the other details about false positives and error correction are noted. I will say that with this particular type of barcode scheme, you can actually resolve a relatively large number of genes just with 3 out of those 4 decode positions. You use the 4th position for an error correction. So you actually only need 3 out of 4 in any particular case to be able to discern a gene from any other gene in the data set with this number of different barcodes available. Among the first times that this was applied was actually in introns, which is a really interesting paper. This was published in 2018. And the question was asked, could we observe nascent transcription within the nucleus itself? So for this particular use case, we're targeting probes to the very 5' leader regions of the gene. And so this part should be expressed in its prior to splicing when it's observed. And after splicing, of course, that region would be expected to be degraded relatively quickly. So when we see this signal, it's presumed to be at an active transcription site. And that's actually borne out by the data, not so much here, but in the next slide. But obviously we see the same kind of signal in terms of fluorescence barcodes lighting up over a series of sequential images. And we see the alignment of those nascent transcripts with specific chromosomal surfaces. Also potentially interesting to note for those of you paying attention at home, in this experiment, they also designed seq-fish probes to long non-coding RNAs, in this case ITS1, to mark nucleoli. And they also combined this analysis in sequence with immunofluorescence measurements of proteins. In this particular case, shown here is a marker for the G2M cell cycle transition. So they could observe which cells were actively undergoing replication in the data set, as well as transcription simultaneously. And then shown here is really the evidence that those transcriptionally active domains that they're interrogating with the intron-seqfish probes actually map to specific chromosomal, achromatic, and surface regions they're mapped by a chromosomal paint type of technology instead. And there's a really high correlation of those two, one and with the other. So we can also think now to apply this type of technology to more of a transcriptome scale gene expression analysis within intact tissues, not for introns, but for expressed and canonically spliced genes themselves within an intact cellular environment. So again, reminding people what the barcoding design is in this particular experiment that I'm about to show, and conceptually in the GIF in the middle, how we think about deconvoluting that barcode sequence. So ordinarily, if we were to look at this large number of genes in a single image, 10,000 genes in a single cell, there'd be a lot of overlaps in XY position of those different genes, one with the other. When we separate out the collection of images with fluorescently bound secondary probes now binding to the primary probes, which are always present, we're able to collect a series of images and separate out those genes optically so that we can observe them in experimental form. And then we can superimpose all of those images back together again to create the full final map of all the genes that are expressed in that single cell in their XY coordinate locations. And of course, we do this by having, again, a well-annotated set of genes that we know are coded for by a specific sequence of fluors, as well as nucleotide sequence positions of the barcodes that were flowing into the system for every given sequence of decode positions in the data set. In practice, this is again in cultured cells, to start with, what was observed is in part shown here in the right-hand side, 3 separate genes as an example. And here we get a sense for the subcellular resolution of the data set, but also even the subpixel or super resolved intrinsic properties of this kind of a data set over multiple rounds of imaging. And the same set of pixels that we're imaging within, we can observe in fluorescent intensity at multiple different image cycles and in almost exactly the same physical XY location. So we can, over 4 cycles, start to get a sense for at a subpixel level, hence the super resolved nature of this data type, where these genes are actually located within an intact fixed cell. The application of that to more of a biological question is started to be shown here, where again, we're looking at 10,000 plus genes in the data set. We're segregating those out into different image cycles and different fluorescent channels. And we're able then, with this kind of a data set, to do a really full single cell RNA-seq type of clustering analysis that's shown in the middle left here in panel E. And as well, we're also able to look at the subcellular localization profiles of those genes within individual cells. So for example, on the oligodendrocytes, astrocytes, or several subsets of astrocytes, we're able to look at specific gene expression patterns and note their physical locations within, in this case, the most of them for these particular examples, the protuberance of the cells, not perinuclear locations, excuse me. I have 2 presentations today already. I'm starting to lose my voice. And then I think the really beautiful part of this data type is shown at the bottom in mostly false color, obviously, but here we have those single cell, clustered single cells, and then mapped back onto the physical locations of those cells and their X, Y, and coordinate positions within the tissue that we just sampled. Along the way, just a quick example of what the quality of the data type looks like. We're obviously collecting a very large number of images, and we're hybridizing and stripping and imaging these tissue sections quite a few times to generate this number of images that we're stitching back together. On the bottom panel is a set of hybridizations for the same probe set from the 1st to the 81st hybridization rounds, and then the composite image is shown in yellow. On the right-hand side there, you see there's a great concordance in the data that we're collecting. So these tissues are very well maintained throughout this procedure. The technical replicates are shown just above that, one seqfish replicate to another with a really high concordance again between those data sets, and also very high concordance with bulk RNA-seq measurements for the genes that we know we're anticipating to collect in this data set, as well as concordance with single molecule fish for at least a subset of those same genes. And then on the right-hand side, again, with the kind of barcoding scheme that we have in this type of experiment, we're actually occupying just a fraction of the available barcodes that we could design probes within. And the intention is to collect 10,000 genes, but they actually have a barcode set that's large enough to accommodate something closer to 24,000. And you see most of the probes are performing quite well. Those are in blue on the left-hand side, sort of like a knee plot for those of you thinking in single cell terms. Again, most of the data in the data sets actually derived from on-target barcodes. And this amount of data is actually several logs greater in count per cell than the off-target that we see. It's a very modest number of barcodes that appear in the data set, between 1 and 10 times across the entire data set. So the background of this type of experiment with this type of barcoding probe design is actually quite low. Just a few other quick examples for what you could do with the data at a subcellular level. When you have it, you can start to cluster genes by their physical locations. And of course, you can do this in a computational way, as shown on the left, but you can also just look at them. And this is a very obviously super resolved image of those spots in a two-dimensional space. But you get a sense for which genes are located in which parts of the cells of interest. And when you look at neighboring cells, I think this is where spatial genomics starts to get really, really fascinating, is this ability now to not just cluster cells and count them, but ask what they're doing in their native environments within that intact tissue section. Here, just as one example, you have a microglial and endothelial cell pair that the authors observed to be co-located next to one another. And within those two cells, the expression of a ligand receptor pair that demonstrate the capability of this assay to define relationships between cells are actually known to be meaningful based on prior work with respect to signaling pathways that are co-located next to one another in the same coordinate system in a very small physical location within a tissue. Okay, just a few more vignettes in the time I have available, and that is to start to think a bit more expansively about how we would apply this type of technology to the entirety of the central dogma. And there's a publication out earlier this year on this work where the authors again performed the RNA-seq-fish assays you've seen described for 70 different mRNAs, seven different RNAs, both fully expressed genes and introns in this particular example. They used a similar approach to look at DNA within the nucleus, and they did so at a number of different scales, or two different scales, I should say, with a number of different regions each. So at approximately megabase resolution, as well as 25 kilobase resolution in nuclei, and then had a series of 17 antibodies that were also used for sequential immunofluorescence, mostly to stain various features of the nucleus, nucleolus, and nuclear envelope. So mostly this paper is really about the interaction between DNA and RNA transcription. First things first, though, a bit of the raw data as you've come to appreciate what it must look like. Some fluorescent dots in space that give us some confidence we're actually detecting and barcoding and correctly assigning specific genetic regions and mRNAs, in this case, within a physical location in the data set. We see a very strong concordance of the DNA-seq-fish results with high C. That's a very confidence-inducing type of result. And then with respect to the epigenetic marks in the sequential immunofluorescence experiment, again, these are mostly designed to assay questions about epigenetic behaviors, properties of different parts of chromatin, whether it's exposed for transcription or not. For example, we see, again, very strong concordance of the imaging analysis for the specific marks with the corresponding ChIP-seq data. So it gives us a lot of confidence, again, that the type of data we're collecting with imaging techniques is actually reflective of the biology that we've known from these particular cells for a length of time now. We can do this again at a full genome scale effectively, depending on the resolution of the probes that we have designed. And we can start to build models of what this expression pattern for different functions and properties of the cell might look like in 2 and 3 dimensions. We have here, for example, a set of the different DNA elements that are present that we've interrogated, as well as a bunch of the different marks for active transcription and epigenetic marks as well within the data set. And what's really fun is when you do something like this and you create a GIF that allows us to sort of wander through the data chromosome by chromosome, and then eventually when we exhaust the human genome, we'll move on to the epigenetic marks and the other protein marks. So we get a sense for where different parts of the genome are that we're interested in interrogating, whether active chromatin or not. It's all obviously packaged pretty nicely in this particular example. We get the DAPI stain followed by epigenetic marks here. And we'll get through some of the fun stuff, and we'll see some of the other kinds of marks for transposable elements in just a moment, as well as laminin. There it is for the nuclear surface. So we get a really beautifully detailed and rich data set that we can now start to imagine applying in all sorts of different ways conceptually to the question of epigenetics and transcriptional activities, as well as how those things actually define cell states in a practical sense beyond what we can observe a gene expression alone, or even a protein expression, thinking back more classically to cytometric analysis. And this is just one example of that kind of work shown here, where we have, again, a heat map of differential enrichment of the individual chromatin markers and proteins, and what those mean for different states of the chromatin that's interrogated here, and reconstructions from that data into specific nuclear zones in 2 dimensions that we can see in the middle panel there, and co-localize on those nuclear zones of active transcription. For example, we can actually start to see the intronic data that we did see in a previous paper, now recapitulated here in this one. And then we can correlate that, that's in the bottom right-hand corner. Now, the state of active transcription in the presence of those introns, not only with the transcriptionally active domains, actually that's the RNA pull2 antibody staining shown just above in the upper right, but we can see the correlation of gene expression of the genes that were profiled, as well as their introns, in association with those active sites of transcription versus sites that were inactive. So in the left-hand column there's a greater degree of variance in gene expression measurement relative to the genes that were not actively transcribed in the same data set. And in the final vignette that I'll have time for today, we're going to one of the most recent publications. This is from just a few months ago in collaboration between Long Kai and John Marioni and others, where they applied seqFISH and single-cell RNA sequencing to mouse gastrulation, and some really, really beautiful work of early embryogenesis to ask questions, again, with the types of technology we have available to us today, how many different cells are present at various different time points during embryogenesis, and where are those cells physically located within a developing mouse embryo, which is really beautiful system of study of rapidly changing cell states as well as cellular locations as the embryo is undergoing gastrulation. It's morphologically moving pieces of tissue around inside the embryo from one place to another place and changing state along the way at the same time. So when you create a single cell map of all of the data that you've collected from both of these techniques, you can cluster that as we're accustomed to in UMAP or t-SNE space. You can use some UMAP in this particular example, and then you can assign those different groups of cells to a physical location in the image data itself. And then you can do fun things like this, where you have UMAP on the one side and spatially resolved seqFISH data on the other side. So you can see, for endothelium, for example, where is that located, what types of gut tissue are present, at what stages of development, and where are they physically located within the developing embryo. And I think this is just a taste for what we can actually think to do in model organisms, as well as human work and organoid work for that matter. There's all sorts of beautiful work that's in front of us now, and we can combine these types of approaches, collect single cell level data sets that have a spatial component inherently as a part of the data that we're collecting. And we can do some really beautiful work to, on the one hand, cluster cells based on gene expression measurements, or we can think about adding in protein and DNA data types as well, if that gives us greater resolution. And we can actually start to visualize where those are in intact tissue that we're collecting from these model experiments. In closing then, I'll just point you in the direction of some data that's on the HubMap portal. This is all collected from human organs, a number of different data sets I'm not going to take the time to wander through today, all available with seqFISH RNA gene expression measurement. And finally close with a few higher thoughts about where we are with the technology that we're developing now, and thinking again about what we're trying to achieve. We're interested in getting single cell resolution, but in situ, and I think we've achieved that. with the flexibility to look at as few as 100 or several hundred genes if you like, but as many as tens of thousands, potentially on the upper end of the spectrum as well. We're capable of thinking also of combining these gene expression measurements with DNA and protein measurement, as well as to ensure that when we look at least the nucleotide level information, we're looking with single molecule sensitivity. And that gives us a subcellular resolution for those things. So we can start to discern biological meaning from where gene expression is occurring or has post-transcriptionally taken those genes within a particular cell state. And we do this with really great specificity. The probe sets, again, that we derive from the annotation are very highly curated, and we spend a lot of time making sure that we have very few or no off-target effects, for example, so that the genes we're assaying are truly the ones that we're interested in. And before I close, I'll just say we've been starting to take some samples for services these days. We have a growing number of capabilities in the laboratory that allow us to think about more and more expansive experiments. So if you're interested in talking more about that, just reach out to us at info@Spatial Genomics. And I will close by saying thank you. And I should have said this at the beginning, it's always a pleasure to see a bunch of friends and be able to share what we're doing and be excited about science together. So hello, friends. It's good to see you. And again, Rong and Ahmet, I really appreciate the opportunity to take part in the series. Now I'll stop here.
Thank you, Brian. Super impressive. I think this is so much work over the past couple of years. So I think folks in the audience, if you want to ask a question, you can unmute. Yeah, Natika, you have a question to ask?
It's a good one. I see it in the chat as well. Shall I answer it, or do you want to ask a different version?
The same version. Please go ahead. Thanks.
No, it's a great question. I was very elusive, but you've managed to pin us down a little bit. So we have undertaken the development of a product platform that takes what occurred in Long Kai's laboratory and effectively creates an instrument with the capabilities that you've seen represented. And we're slowly but surely adding to the repertoire of things that the instrument can do. We're currently using it only in services and for our own internal purposes. At the moment, we're looking at having something that's widely available as a product next year, but I won't be able to say much more about that at this point.
Yep, thanks.
Yeah, I think my laboratory has been kind of doing sequencing business for years. We're not good at imaging. So in this case, how challenging it is to do the image analysis, or you're gonna kind of democratize that as well by offering some kind of computational image processing software. And, but I think it's going to be very CPU or GPU heavy computing, right? So yeah, what it can help us if we try to adopt it.
Absolutely. I mean, that is one of the parts of the platform that we have to consider, and we're working very hard. Actually, arguably the most amount of effort and resources is going into the software side of it right now. And there's some really great people there at the company that are working really hard on this problem. But we're collecting a series of images. We're collecting a series of fluorescent spots within XY positions in those images, aligning all of those back together again. So all of this is largely occurring during the run of the instrument itself. So it's actually kind of funny for those of us who've been around long enough in genomics, what was old is now new again. So like the genome analyzer many years ago was pumping out what was then giant stacks of image files that we had to deal with computationally and turn that into a CSV file effectively. We kind of have the same problem with even more data, and we're solving it. So the good news is that the full platform will have that baked in capability to manage the image analysis. So the amount of data footprint that's landing on your local server or a cloud-based one will be a manageable volume of data. But beyond that collection of data, we have to deconvolute the barcodes of the fluorophores that we're capturing and convert that into a matrix file of genes by cellular locations and include actual subcellular or pixel by pixel location effectively. So I like to think of it as containing the same kind of information as a single cell data set, but with the added resolution of a location within an image that we know from just empirical observation of the genes that we've captured in the data set. There's other parts of it that I haven't really talked about, but obviously we have to segment cells. And so we need to know where the boundaries of those cells are. There's a variety of different ways to do that. We can incorporate one or more of. We have looked at a couple different things. I'm going to have to tease you on this part. We're not going to show any segmentation data, but we do know that we have to do it. And there's a number of different ways to do it, depending on the complexity of the tissue, right? There's some things that are very regularly organized that are quite easy, and then a bunch of other tissue types that are much more challenging. So most of that work will either occur on the instrument or shortly thereafter, after the run completes. It is GPU intensive, but it's manageable within current computer configurations. There's nothing particularly magical about the kinds of computing required these days to manage these kinds of workflows. And then beyond that, there's you can imagine taking this data. It is single cell data in a sense. It has a spatial component to it, but you can extract that away and cluster cells as you would with any other analysis. And then the beauty of it is you can assign those cell clusters and states back to the physical locations they originated from. And so there's a visualization component to that, and a computational and visualization components to that are also required. Hopefully that gets to most of your question. I do see another one in the chat from Charles. I'll go ahead and take a swing at this one. We were expecting this because everybody's technologies this is the question that seems to be asked the most in most of these presentations. How is it conceptually similar to other technologies that are in this seminar series? Again, I'll have to be somewhat circumspect here in answering the question, Charles, because the simple truth of the matter is that neither we nor anyone else has been fully transparent about how they're actually doing all of these different types of analyses. I will say that, again, I hopefully was able to describe it well enough that you can be convinced that we're using microscopy and a barcoding strategy that takes fluorescently labeled oligonucleotide probes and uses those to assign physical locations to genes that are expressed in a tissue sample of interest. But beyond that, it's difficult for me to say specifically, or more specifically, how similar or different it is to anything, let alone one particular technology. Lou's asked the question, is a box like in the roadmap? I did get to that with Nashiket's question, I hope. It's in the roadmap, yes, of course. We're just not going to say quite when yet, but stay tuned. It's all hopefully not too far off in the future.
Brian, this is Sean, by the way, from BioTuring. So this is, I enjoyed the talk so much, and I want to add to Rong's questions about, do you intend to democratize the data part so that you can put and I'm talking about currently, do you put any raw image data out there so that the hungry computational people can, contribute to that as well?
The rawest of raw data is from this type of an experimental analysis so massive, it would be very challenging to even keep that anywhere for any length of time. So in one way, no. I mean, the system is designed to actually take all those images and sort of stack them together. At least intuitively, that's how I think of it. The computational biologist here will probably tell me it's slightly different than that. But conceptually, we have to do that on the instrument itself while it's collecting this data, or it would destroy whatever server you're sending it to. So that level of data is what's passed along at the end of the analysis. So an image file with, again, the coordinate locations of all the genes that are captured in the data set. So that's the rawest of data that we could hope to provide, I think, in a practical sense. I don't think that's going to be dramatically different from other platforms in this regard at all. I think we're all kind of coalescing around the same kind of data output.
Right. I meant the image file for cell segmentations and other parties, are they available?
Yeah, the intention, I should say, I guess to follow up on that question a bit more specifically, while we think our segmentation is good, I don't think we're ready to say, I don't know if anybody in this room would be ready to say that their segmentation is perfect. So I think we will expect to play with segmentation manually to some extent, depending again on the complexity of the tissue. Some of them are very straightforward and others are a bit more challenging. So that kind of analysis, I think, has to be supportable, at least for the foreseeable future. I think we can all imagine a place, though, where we get sophisticated enough with this kind of analysis and practiced enough and empirically experienced enough that we can start to think about segmentation occurring automatically. But In fact, that would happen anyways. It's just the quality of that result is probably something that would be open to interpretation for some time to come. I wouldn't want to speculate, but within the next couple of years, let's say we've collectively solved these kinds of issues, so we don't have to worry about segmentation any longer most of the time.
Yeah, but on the other hand, this is an opportunity, right? I think Sang asked this question. I sense that's so if you can kind of share the image data, actually it's a great opportunity to the computational community. Actually, a last colleague, Dave Valent, who was one of the speakers last year, and he has been using like the crowdsourcing and all those kind of gamification approaches to do cell segmentation. It's kind of manual, but you get thousands of people to help you manually segment. I think, yeah, if you have the image there, I think lots of interesting things may happen. Just want to point out that that could be very, very beneficial and a great thing to do for us as a whole community.
I agree, completely agree, completely agree with this.
Yeah, I don't think there's any real disagreement on that point. It's just a question of how raw is raw, I think, to answer your question explicitly, right? Because there's only so much data we can actually produce that, or up to a limit rather, that's supportable by most computational platforms. We don't have all the power of astronomy and all of the world's server farms at our fingertips to manage every individual data set. So I totally agree with you. I think there's a lot of room in this space for computational development, and certainly that's the way that genomics has always been, right? There's always been a lot of tinkering around the edges at the software side especially, and I think this is where the beauty of all these data types inherently is really fully realized by having a community of people that can churn away at different solutions and problems. And I think that's ultimately what our platform is designed to support too. I don't see us behaving dramatically differently there. But again, if you can get to a point where the industry agrees that the problems have been solved and there's a good way to do that, then we can move on to different problems.
Brian, agree. 15 years ago, FastQ was big. Now image is big, right?
Yeah, I agree. Everything that's old is new again.
Yeah, actually, that's a good point. I think based on the raw data of Illumina sequence images.
Oh, that's right. Yeah. Also, agree. So Brian says, okay, open to what extent, draw to what extent. Like we say, okay, Illumina, the draw file, okay, the image file of Illumina is not yet, we don't see it. Okay?
Well, there was a period of time very early on where people actually did have image files back then, and they tried to do a better job of cluster definition than Illumina did. And I don't think anybody actually really achieved that systematically. And there wasn't a need for it, right? The company innovated faster than that, and it didn't really require that as an industry. So will we repeat that same kind of feat with segmentation, for example, in spatial genomics? Possibly. But the future is a big place. Let's all wait and find out.
Yeah.
Right.
There's another question that I would just take a quick second to answer, because I realized I didn't answer it earlier in terms of the product platform or even the service platform for that matter. It's not just about the hardware and the software. Obviously, there's a third component to this, and it's pretty obvious for the entire genomics community. There's reagents involved, and there's probe sets, right? Because we're looking at specific probes that have been curated again to detect whatever we're interested in detecting. So the answer to that question is really we are looking at developing a set of standard assays. Obviously, there's relatively canonical probe sets that people have been using across the genomics industry in one way or another for quite a while now. We're not deviating greatly from that, but there is a lot of opportunity for semi-custom or fully custom content to be added to either adjacent to that set of standard content or completely in separation from it. So we are trying to support as much of that as we can. Obviously, there is a practical limit to how much of that you can do, but we do think that, again, this is where a lot of the interesting science happens, is not from us being prescriptive about what content we think is interesting, but enabling the content that the community thinks is interesting as well. And so we'd like to be able to support as much of that as possible.
I think most of the data you showed today from those publications are, I think a brain tissue, that's one. I think other tissue types, most healthy, normal tissue. So have you sort of collected data for like a disease tissue types, and also whether or not it's possible to look at something like a copy number variation and mutation landscape and those kind of disease-related genomic alterations?
Yeah, great questions, Rong. And there's a couple different ones in there, so I'll try to answer them all in series, and hopefully I don't forget one along the way. But yes, we in the lab's history, we've started to look at a number of different tissue types. There's obviously a number represented in the HubMap data set that I referred to as an example. It's not all done in cultured cells, obviously, or even in brain. There's a bunch of other solid tissues represented. We are starting to collect a large amount of data on a number of different tissues in our own hands, and through service relationships, we'll get our hands on additional data sets for some research areas and diseases. We won't always be able to share that, obviously, because that belongs to somebody else for publication first. But sure, yeah, we are interested in doing that as much as we can. The challenge that we have as a commercial company, obviously, we're not physically co-located next to a clinical center. So it's a little bit more challenging to get that full diversity of disease states that you might like. Let's see, there was another question. Of course, I've now forgotten it, Wrong. What was the next set after that?
Yeah, I think like a genomic alterations, like a CNV
of course,
and mutations.
Yeah, I think these are tractable kinds of questions. And I say that with a bit of confidence because in the original single molecule FISH papers from Arjun, for example, he demonstrated the capability to assay single nucleotide variants. And so I think there's a lot of really interesting work to be done there. It changes the signal to noise calculations or ways that you detect things a little bit maybe, but while I think those are a little bit further away from us in a practical sense, because each of those questions starts to get a little bit more defined for individual research projects, I think technically they're tractable problems that we can solve. They have been solved. We just haven't implemented them as a service component yet, for example. Mm-hmm. And then Lou's got another question about frozen or FFPE. I think FFPE, again, is a very common question, and it's certainly something we're expecting to be able to support. And I think that's the beauty of these probe-based technologies. Like seqFISH is not inherently limited by RNA fragmentation. It's just a series of probes. In fact, it's actually probably more robust in that respect. As long as the RNA in those tissues is not so badly degraded that it's basically all shredded to tiny, 10 nucleotide pieces or something, we should be able to detect it. We are working on refining the probe sets where we feel like we need to be able to assay FFP more confidently, but I think inherently that's a sample type that we can access depending, again, on the quality of the tissue, maybe the type of FFP we're talking about, et cetera. But I feel like that's something that we'll see more of in the future.
Yeah, I think you talk about the RNA fragments in a FAP sample, so kind of along the same lines. So do you think for the very small genes, or not small genes, but like the small RNAs, do you think that's possible to look at using seqFISH like a microRNAs or other non-coding RNAs?
Again, microRNAs might be a slightly more special case because the actual real estate there for primary probe hybridization is much shorter than some of the other probe designs that we could imagine. I'll hesitate to say absolutely yes in every case, but it might be possible to do some of that work. But shorter genes, sure. I mean, we tend to have a pragmatic cutoff in terms of how many genes we would like to have to be able to detect something, but you can always, I think, at least as one example, you could have a shorter gene, let's just call it a couple hundred nucleotides in length if you like, but as long as the probe designs are quality and you know that they will hybridize effectively, and you'll be able to do that by itself as a serial hybridization and imaging later in the run versus barcoding it with something else potentially, because it might be a question of signal to noise. So I think there are, again, some things that we can do for smaller genes. Again, this is not terribly dissimilar to single nucleotide variants where you might have just one probe that actually occupies the space for that particular assay. And here we actually have the luxury of having several to occupy a couple hundred nucleotides of imageable space. It's just not the first thing that we're working on. We are trying to get the full transcriptome kind of data set size of gene panels identified, and then we can work back into the sort of the edge cases around the data set. There's a question from Roy that I'd be able to address here briefly. There are some publications, I didn't really get a chance to go into the details of them too far, but in terms of detection, sure, are there different ways that you can use secondary probes? And in this case, HCR is referring to an enzymatic enrichment of the signal, sort of signal amplification method, if you will. We've certainly given some thought to whether we need to do that. And right now we're trying to avoid it for the first sets of content that we're developing, because it's just pragmatically easier not to worry about added complexity in an experimental system like that. But I think, so while it's not necessary and we're not using it by default, there may be some use cases eventually where some kind of additional signal enrichment is required or interesting, depending on the types of things we're trying to detect. But I do think that while that was an interesting technological development for the publications in which it was present back in prior history of just a couple of years ago, there's a few things that we can do now that are a bit easier that don't require us to do that to detect the same kinds of genes of interest. But thanks. I mean, somebody's aside from Guo Chen, there's someone who's actually read these papers in a bit of detail.
Reward.
Thanks, Joe. Yeah.
Yeah. Also, I was very impressed with the data to probe the intron region of the RNA. So then, I haven't seen anyone doing anything kind of similar like that using fish. So in this case, actually it's on a kind of whole genome transcriptome scale. So in this case, you are able to see kind of nascent RNA and all mature RNA. Do you think you can kind of look into this temporal aspect, very similar to RNA velocity? Can you do something like RNA velocity using the sequential fish?
Yeah, I think that's a really beautiful question. I think the idea of doing pseudotime or other kinds of projections like that in tissue is a fascinating idea. It's funny that you mentioned that, because if you look in the introduction of that Intron paper, you'll see just a simple note in reference to Lamano's publication, which I'm guessing had just come out in enough time for that note to be made part of the intro as part of the revision process or something. So they did realize it was present, but they didn't have time then to go back and redo any of the analysis, at least that I've seen. So could you imagine doing that with a combination of intron seq fish and standard genetic seq fish? Why not? I mean, I think it'd be really interesting to try. Do you get the same information that you get from a single cell RNA sequencing type of approach? I'm not sure, right? Because well, it depends on how you design the probes. Let's put it that way. If you just took the set of probes that we have for the 5' leader from that data set, you might see something subtly different than you would get from an RNA velocity experiment of single cell RNA-seq data, where you might have a longer transcript that you've ultimately captured with more 3' introns present or something of that sort. But these are the fun technical details like we get to play with when you have the data available finally. So I'm looking forward to somebody doing that eventually if we don't get to it ourselves.
Yeah, we do have quite a few computational folks in the audience. Yeah. They probably can go ahead and take a look.
There's so much fun in this space, right? There's so much. There's so many fun ideas to play with. I'm looking forward to somebody getting to them eventually. Yunshen Yang has another question: in which genes are helpful for improving segmentation as opposed to intracellular patterning? In segmentation, especially, I think it's easier to think about protein analysis of the cell surface than genes themselves. I mean, you can certainly get segmentation through the gene expression pattern itself, and in a way, you can look at. You can imagine looking at gene expression in aggregate and saying what genes are sort of co-located with each other and with DAPI, for example, And does that pattern make sense relative to some kind of prior atlas of cell states that you're familiar with? I think that's a bit more complicated, honestly, than you really need to, but you might use it as you can imagine using that to validate segmentation results to some extent, I suppose. But I just feel like immunofluorescence is so much more straightforward as a way to address that question of segmentation, that it's certainly the easier and more systematic thing to do first.
Okay. So if no more questions, yeah, let's thank Brian again. This is really terrific.
Well, thanks everyone. It's been a pleasure. Thanks for all your questions. Those of you who I know, it's good to see you again.
Yeah. I think next week we're gonna have Dr. Inju from Pacific Northwestern Laboratory to give us a talk about single-cell and spatial proteomics. Those are mass spec. The technologies he developed is called nanopods, and hopefully you will come back again next week. And thank you, and have a good weekend.
Thanks, Ron. It's been a
Thanks, Ron.
pleasure.